Welcome everyone. Today we are going to learn about nerve impulse propagations. This is my introduction video. At first we will learn what is nerve impulse propagation and then some important facts. Nerve cells or neurons transmit electrical signals that propagate along the length of this structure. This is a typical structure of a neuron. It contains cell body and projections. These are called dendrites. And this is action. The length of action can vary from 1 millimeter up to 1 meter. And the end of these neurons is called axon termini. Now the axon termini of one neuron can make contacts with dendrites of other neuron, and the junction is called synapse. Now this red line indicates the direction of nerve impulse or electrical signals. Remember, this direction always from the cell body through the action to the action terminus. That means it is unidirectional. The reverse is not possible. The reverse direction is not possible. Like the nerve impulse could not propagate from axon termini through the axon to the cell body. So remember, nerve impulse propagation is unidirectional and it propagates along the length of this neuron. Now the first important point I want to mention is excitable cells. Neurons are a member of a class of cells called excitable cells. Now not only neuron but muscle cells, beta cells of pancreas, eggs, these are all excitable cells. Now this excitable cells has two important properties. The first is they can sense the change in membrane potential. I have written here, membrane potential is also called electric potential or voltage gradient. Now let me explain what is membrane potential or electric potential or voltage gradient. Suppose this is the plasma membrane. Now we know that plasma membrane surrounds a cell, so it demarcates the boundary of a cell. The outside is called exterior or external environment and the inside is called cytosol. Now if you insert a microelectrode inside this cytosol and if you keep another microelectrode outside of the cell and if you connect these two microelectrode with a potentiometer or voltmeter, you will get a reading. That is, in normal cells or resting cells like resting nerve cells or resting muscle cells, you will get approximate value of minus 70 millivolt. Now this value is the membrane potential of the cell. Now in case of neurons or muscle cells or beta cells of pancreas, they can sense the change in this potential. Now this potential can rise or can fall, and these cells can sense the change in membrane potential. So this is the first fact, that these cells sense the change in membrane potential. We also can call it voltage gradient. Now when they sense this, there is a change in the membrane potential, they perform, the second point is important, they perform effector functions. Like in case of neurons, they propagate the nerve impulse or electrical signals. In case of muscles, they contract, and in case of beta cells of pancreas, they secrete insulin in response to membrane potential change. Now let's talk about the second important fact, that is electrical signals. Neurons can generate a variety of electrical signals, and we categorize these signals into three. One is receptor potential, second is synaptic potential, and third is action potential. Now what is receptor potential? Receptor potential is due to the activation of sensory neurons by external stimuli like light, sound, heat, touch, pressure, or even chemicals that excite the taste buds. Now I will give one example of receptor potential. This is one example of receptor potential. If you touch the skin of a finger, it activates Pacinian corpuscle, and if you record, you will find a change in membrane potential. Initially it was minus 60 millivolt and then it rises. In case of synaptic potential, if you activate the synapse, you will find that there is also a membrane potential change. Initially minus 70 millivolt, then it rises. And this is example of action potential. Both the receptor potential and synaptic potential can generate a current that can activate or generate action potential. Also, artificially in the laboratory, you can insert a microelectrode inside a neuron, and if you connect this microelectrode with a battery, you can generate a current, and then you will record. If you record, you will find a change in membrane potential, and this is called action potential. There is a difference, as I have mentioned, the amplitude of the action potential is different with the receptor potential and synaptic potential. As you can see, the peak of this action potential can touch plus 40 millivolt. In normal condition, receptor potential and synaptic potential can generate electric current, and that electric current can generate action potential. Also, in the artificial condition in laboratory, we can insert a microelectrode inside a neuron and can connect the microelectrode with a battery. Then we can generate an electric current and also action potential artificially. Now, action potential performs some effective functions, like it helps in transmission of informations in the central nervous system and peripheral nervous system. Also, it helps in the skeletal muscle contraction. It controls timing of heartbeat, peristaltic movement of gut, contraction of uterus. Now let me talk about the third important fact, that is ion flux or ion movement. See, all of these electric signals are due to the ion movement through the ion channels. Now there must be some force for this ion movement, and we divide this force into two categories. One is called chemical force and another is called electric force. The chemical force is also called ion concentration gradient, and the electric force is called membrane or electric potential. As I have mentioned earlier, what is membrane and electric potential? Now, ion concentration gradient means there is a difference in ion concentration inside and outside of a cell, because plasma membrane separates the exterior and interior. So there is a difference in the concentration of ions that generate ion concentration gradient, and there is a voltage difference in inside and outside of a cell, that is called membrane or electric potential or voltage gradient. Now together these two forces called electrochemical gradient or ion motive force. See, as because ions contains charge, so they are also influenced by the electric force. Hope this video will help you. Thank you for watching and always keep learning.